6.3 Enzyme. Most metabolic pathways involve many small chemical reactions. Each of these chemical reactions in our metabolic pathways is controlled by a specific enzyme. Enzymes generally are proteins. There are a few enzymes which are RNA molecules, but generally we can consider enzymes to be proteins. We also call them a biological catalyst. This is something that is going to speed up the rate of a reaction. So why do we need enzymes? Here's one of the energy diagrams that we looked at in the last segment. This was our exergonic reaction. And right in the middle, there's something that sometimes I call the speed bump. This big hump in the diagram, it's called the energy of activation. This represents the energy that must be present in a system in order for the reaction to proceed. So energy is present in all systems. Molecules are constantly moving around, bumping into one another, and these reactant molecules cannot react unless they run into one another with sufficient force and also in the correct orientation for the reaction to take place. If we were in the chemistry lab and we wanted to speed up a reaction or even get a reaction going, we would need to supply energy to get over the speed bump, and so we could simply put our reaction on a Bunsen burner or on a hot plate. However, that's simply not an option for living organisms. So living organisms need some other way to overcome the speed bump. We have to somehow get past this energy of activation, and this is what enzymes do. Enzymes are going to lower the speed bump so that the reaction can proceed at physiological temperature. I like to call enzymes the molecular matchmakers. They are going to get their reactant molecules and they bring them together in just the perfect orientation, in just the right environment, so that the reaction can proceed. Here's a simple little diagram that shows an enzyme. Enzymes are generally proteins. They are folded up into a very specific shape, and this creates something called an active site. The substrate is the fancy name that we use for the reactant molecule. The substrate molecules can fit perfectly into the active site, and that places them in the correct orientation for the chemical reaction to take place. So the substrate binds to the active site, and then the product is released from the active site. There's an equation down at the bottom where E equals enzyme plus S, which is the substrate. We get the enzyme substrate complex, where the substrate is attached to the active site, and then we release the product. Now the enzyme is unchanged, and so enzymes can work many times, some of these many times per second, and the enzyme is unchanged in the reaction. So here is another one of our energy diagrams. You see the blue line represents that high speed bump, the energy of activation, when the enzyme is not present. And if we have the enzyme present, that lowers the energy of activation, again, because those substrate molecules are positioned in the correct orientation and environment for the reaction to take place. Enzymatic reactions can be degradation reactions, so this would be catabolic reactions, most probably an exergonic reaction where we have a large substrate molecule binds to the active site and we are going to break some chemical bonds, and so we release a smaller product. Again, the enzyme remains unchanged. We can have a synthesis reaction, so this would be an anabolic, probably endergonic reaction, where multiple small substrate molecules bind to the active site. We're going to form new chemical bonds to create a larger product. This diagram shows what an enzyme might look like. Most of them are proteins, which are chains of amino acids, and so the little small dots represent the individual amino acids. Proteins must fold up into a very specific active three-dimensional conformation. Whenever they do, that creates this active site, which is where the substrate is going to bind. Now, it's not quite like a lock and key. The current model is called an induced fit model. But the active site still has a specific shape. Only a specific enzyme can fit into that. A specific substrate. And then the enzyme changes conformation just a little bit. This is the induced fit model. And that allows the specific reaction to take place in the active site. Enzyme names, many times we will take a portion of the substrate name and we put the suffix ASE on it. So for example, if the substrate were lipid, a fat molecule, an enzyme that breaks that down would be a lipase. Lactose, which is a disaccharide milk sugar, the enzyme that breaks that down is lactase. Incidentally, people that are lactose intolerant cannot digest lactose because they do not have sufficient amounts of the enzyme lactase. Proteins, this enzyme that breaks down proteins, again, we can use a portion of the substrate name with the ASE. So a protease is an enzyme that breaks down proteins. There are a number of factors that will affect enzyme activity. The first of these is temperature. If we look over here on the diagram, this is showing the rate of a reaction at various temperatures. And so we have this bell-shaped curve, and the top of the curve would be the optimum temperature for that enzyme. As we get colder, when you decrease temperature, all molecular movement slows down, and so that would slow down the rate of the reaction. However, if we get too hot, very high temperatures will change the shape of the enzyme. If you change the shape of the enzyme, that alters the shape of the active site, and so the reaction is going to slow down. So generally, we can say increasing temperature increases enzyme activity. However, if we get beyond that optimum temperature, it's going to slow down the enzyme activity. If we get it too hot, it will destroy the enzyme. This is called denaturation. The protein will completely unfold, destroy the active site, and destroy the activity. pH also influences enzyme activity. Every enzyme has a specific optimum pH. In this example, again, we have the rate of the reaction, but this time it is plotted at various pHs. Pepsin is a protease, breaks down protein. It is found in the stomach. The pH of the stomach is about 2. So if we look at the rate of pepsin activity at various pHs, it should be no surprise that its optimum activity is around 2. Trypsin is another protease. Trypsin is found in the small intestine, so we have a separate bell curve for trypsin. Looking at the curve, can you see what the pH of the small intestine might be? Peak activity for trypsin is about 8, and that is in fact the pH of the small intestine. If we take pepsin and put it in the small intestine, it won't work. Or if we take trypsin, put that in the stomach, it won't work. Again, these have specific pHs where they are functional. Substrate and enzyme concentration. Obviously, if we change the concentration of the substrate or the enzyme, that's going to influence the rate of the reaction. And some enzymes require cofactors. These could be vitamins, they could be minerals, little small molecules that assist the enzyme, and if these are deficient, the enzyme activity is going to be affected.